Okay, perfect. Okay, so thank you, everyone. Our presentation tonight is titled Wolbachia and its insect manipulations. Our presenter is RJ. RJ gave a talk to our chapter last year. She is now a third-year PhD candidate in the Ware Lab with the American Museum of Natural History's Richard Gilder Graduate School Comparative Biology program. Her work focuses on the systematics, phylogenetics, and host-parasite associations of the twisted wing parasite Strepsiptera and their enigmatic placement in the insect radiation. In the course of her dissertation work, she is using more than 24 million preserved invertebrates housed in the American Museum of Natural History as previously untapped resources of archival genetic material and ecological data to inform questions about Strepsipteran evolution. RJ is originally from California and first began her research career as an entomology major at UC Davis. Fascinated by parasitoids, she observed taking over butterfly cocoons. So with that, RJ, I turn it over to you, and I will be right back.
Thanks, Jenna. Hi, everyone. It's nice to be back. Thanks for inviting me back to give a talk. It's always a fun time, and I'm excited to give this one because it's a very new material to me, but it's very relevant and exciting to my research at the moment. I am not someone who's previously worked with Wolbachia, so what you're seeing is like what I've been learning in the past month or so. And I think it's really cool because I love parasites, and then having these extra multi-trophic interactions on top of parasites or in combination with parasites, I think is really takes the cake. So let's get started. Okay, here we go. So I'll talk to you in about, or this presentation will be in about 4 parts. First, we'll go over what Wolbachia actually is, and then what we use it for in our daily life, well, not in our daily lives, but as humans, how Wolbachia has affected insects, especially butterflies, seeing as that this is a butterfly club, and then what I'm doing right now with Wolbachia in the American Museum of Natural History for my dissertation. So most of the presentation, especially this background, is going to be drawn from these 3 papers. This research letter about species infected with Wolbachia. Wolbachia is a bacteria, but we'll go over that. I just keep wanting to say bacteria. This review, which covered a lot of material and is a really great resource for learning more detail about Wolbachia. And then this paper, which was very helpful for mechanisms and transmission. So what exactly am I even talking about? We'll talk about the overview, the history, and its mechanisms. Wolbachia is the genus name for a gram-negative bacteria, which means it doesn't stain with a gram stain, and that's classifications of bacteria. It comes from the order Rickettsiales, which is a pretty big group. And it is the most common parasitic microbe in animals because arthropods, insects and arachnids and other small groups, invertebrates, are the most numerous animals on this planet. So therefore, this is the most common parasitic microbe in animals in general. It's notable for its reproductive manipulations. That's what kind of makes it so well known and fascinating. And it's found that it's the most common reproductive parasite in all animals because it will affect the reproductive systems of its hosts in really interesting ways. A recent meta-analysis paper also estimated, one of the ones that I referenced in the earlier slide, that over 65% of insect species harbor Wolbachia, this bacteria, which makes it the most abundant intracellular bacteria genus so far discovered. So it's across at least 106 insect species, but that's only what we've been able to see. It's probably in a whole bunch of them, since we're not able to test everything at the moment. And these associations can be very, very complex. This is what it looks like, courtesy of some Wolbachia mosquito studies and one of the first electron micrographs. So this phylogeny, this image, is from the Cell Host and Microbiology review titled Living in the Endosymbiotic World of Wolbachia, a Centennial Review. So this was done in 2021 and it's a really good summary of everything to date. So the left-hand side, figure A, is where Wolbachia currently stands in bacterial tree of life, the bacterial tree of life, and within its own group. And then figure B is a summary of its supergroups, because Wolbachia, as we refer to it, is technically only one described species, but that's up for debate. There's a whole discourse around if it's one species or multiple. So for now, we treat Wolbachia as having supergroups. And those supergroups are defined by who they infect and what they do, mostly what they do, but that's becoming more of a gray area since people are finding that some strains do different things in different insects or invertebrates. I like this figure because it can show you visually just how big or maybe not how big, but how much work has been done on some of these groups. We can see that A and B are the largest supergroups for Wolbachia, and that's because that's where the most work has been done, since it's where insect interactions and our human use of Wolbachia takes place. But they also infect things like nematodes. They're actually beneficial in nematodes. They help nematodes. Spider mites, pseudoscorpions, and other arthropods, and even scorpions. Pseudoscorpions are different. Okay, so sorry, I keep wanting to cough, but I want to. So elaborating on the debate in the Wolbachia field as to whether all of them should be given the one species designation or whether different species nomenclature should be applied. Until this issue is resolved, the convention has been to just refer to this bacteria as Wolbachia, the genus, with strain designations, as I mentioned. So there's this database that exists to compile all sequences of 5 genes and a couple more, but there are 5 genes that are currently the standard for multi-locus type sequencing or sequencing types, which is MLST. That's why pub MLST is up there. And you can plug sequences into this database or multiple sequences or a whole genome, a whole DNA strand, to see what genes in which strain profiles are a match. So that if you have something that contains Wolbachia, you can try to identify what strain it belongs to or what other insects or arthropods might be infected by it, to try to understand what your Wolbachia might be doing. And that's what this looks like. So you would just paste in a Wolbachia gene locus, which I think is very neat, and I'm happy that that exists right now. Oh, I forgot to time myself. So I'm just starting that now, and I'll just assume that I'm like somewhere around 8 minutes in. Sorry. So here's a timeline. I thought of that because I saw the word time and the paused clock in my presenter view, but let's go over the timeline. Until the early 1990s, Wolbachia were considered to be members of a rare and inconsequential bacteria genus. They were first discovered in 1924 and placed in a different group, and then all that taxonomy got sorted out. Then in 1950, Culex mosquito cytoplasmic incompatibility was discovered as a separate phenomenon. They didn't know at the time that it was associated with Wolbachia. But cytoplasmic incompatibility occurs when, now we know that it occurs when an infected male mates with a female that is either uninfected, or infected by a different strain. So they won't be compatible. They won't make fertile offspring. So in essence, if the male is infected by a Wolbachia cytoplasmic incompatibility inducing strain that is non-existent in its mate, then it's an incompatible cross and they can't have offspring. So this was exciting for vector control implications for the control of mosquitoes. In 1970, the Wolbachia part of it was realized due to antibiotic curing as an experiment where applying antibiotics to the mosquitoes resulted in them being compatible, and people realizing that, oh, it's a bacteria in these mosquitoes that's making them incompatible with each other. And that one experiment, the antibiotic curing, has become the standard for experiments determining if Wolbachia is causing reproductive issues in all systems that are working on Wolbachia. Excuse me. Then it was spreading. By the 1990s, there were discoveries of cytoplasmic incompatibility in several other major insect orders, and molecular methods started being applied to those to see if these were the same bacteria or if there were other bacteria or what kind of evolution was going on in this system. Then by 2010, the first sequenced Wolbachia genome came from the fruit fly, Drosophila melanogaster, and a lot of implications came from that on how Wolbachia evolves, what exactly it's doing, and why it's different from everything else. Then in 2020, which we are in now, there's widespread typing with multi-locus sequencing typing, multi-locus MLST, and the database that exists to compile everything and facilitate research. And now there are also health applications and active projects going on really since the 2010s in vector control and integrated pest management. So elaborating on those odd features, they have very small genomes. That's actually about the same size as a strepsipteran genome, which I can talk about later. It's very interesting, but technically they're both parasites, and small genomes are very indicative of parasites, or they're typical of a parasitic lifestyle, because you can lose all of these genes if you're not really using them, since you depend on your host for like all these things that you might need to do independently if you weren't a parasite. So that's neat. But then researchers found that there were a high number of mobile and repetitive elements in Wolbachia and the Wolbachia genome, which is very different from other obligate endosymbionts. Usually they're very compact and there's not really that much that you wouldn't understand like the function of. But in Wolbachia, it's not really known what these elements are doing. In general, we don't really know that much about repetitive elements. They are more than 14% of the genome, which is a relatively high number, repeats. And a very neat thing, I think, is that these encheiron domains or ANK domains exist in the genome, which are virus-like elements. They're common amongst eukaryotes, but unusual in bacteria. And they usually mediate host pathogen protein interactions. They're bacteriophage sequences, bacteriophage viruses, and some of these phage elements are actively transcribed and expressed in the Wolbachia. So this implies that there's a fourth player in these interactions, a virus. So that makes it a quadripartite interaction where a virus might be influencing the Wolbachia, which might be influencing an insect parasite, which is influencing its host insect. And I think that's wild. So that is also pretty new, but you could see in the image in the figure that I showed you earlier that these are where we know these little bacteriophage icons are where we know those genes, those domains exist in these strains of Wolbachia. And we don't understand what the implications are quite yet. But it might be really cool. So to talk a little bit about their transmission and how they get into different hosts or get into the rest of the population of their same species, we can talk first about, let's talk first about vertical transmission because that is what is required for all Wolbachia to pass down into next generations and to survive within a population. Essentially, Wolbachia is inherited through the female germline, so through mothers. They pass through the ovaries to developing eggs, the somatic tissue. Yes, and only in females. And then they proliferate through offspring. So you can imagine that it's more beneficial for them to have more females in the population. Excuse me. Horizontal transmission is interesting because this might be what caused host switches, what caused it to spread through the insect tree of life, or even pods to insects. Sorry, insects are arthropods from crustaceans or from arachnids. That's what I was trying to remember. Arachnids to insects or vice versa. And this can happen through cannibalism, cannibalism or predation, but this seems uncommon since the microbe would need to survive digestion, which is a pretty arduous process, I would think. And then shared niche, which applies to microbes that can survive either within or on the surface of food. So some Wolbachia strains can actually survive extracellularly for up to a week and up to 50 days for some if they're in leaf-like vessels, phloem vessels. And this would be where one species would leave the Wolbachia on the food and then another species would come pick it up and bring it into their system. The most common probably is through parasitism. Host-parasite shifts seem most common, and that's because the physical association between a host and parasite typically lasts longer. It occurs at various developmental stages, and it enables Wolbachia to directly contact a lot of different host tissue. So that sounds reasonable. But given all of this, it's not a sure thing. Even if a new species picks up the Wolbachia from something else, it has to still go through vertical transmission through a mother to her offspring in order to survive and spread in that new species and establish itself. I also want to mention these mechanisms of reproductive manipulation, which I think are absolutely fascinating. So once they're in these groups of insects or in a new host, in a host, they can cause all of these different wacky reproductive phenomena. There's parthenogenesis, which is where all female offspring come from unfertilized eggs, essentially clones of oneself. And this is a natural form of reproduction for some organisms, but not for a lot of them, such as Hymenoptera. So bees, wasps, and ants, Wolbachia can cause, instead of eggs turning into males, unfertilized eggs turning into males, they'll still be female. And then there's also pathogen resistance, which has implications for vector control, but it can reduce the pathogenic viral loads and increase the survival of different insects, and it was first seen in Diptera. These aren't the only groups, by the way. These aren't the only orders that these phenomena are present in. This is just where it was first recorded. And then feminization is a very interesting one that we'll talk about more because it occurs in Lepidoptera. And this is where Wolbachia infected males will develop phenotypically into females. And this was first found in isopods or woodlice or roly-polies. And then mutualisms are where both partners benefit, the Wolbachia and the host benefit. They can either do this through increased metabolic benefits or reproductive benefits, facilitations. And this happens in filarial nematodes, which has also big impacts on human health and disease. So how do we use Wolbachia, speaking of human health and disease? We use them for pest control. To say a little bit, I suppose. Wolbachia can be used to manage outbreaks of dengue and malaria. It was found in Culex mosquitoes, and essentially introducing a life-shortening Wolbachia strain to mosquitoes, which don't naturally have that Wolbachia strain, can eliminate the older insects that contain more parasites since they accumulate over time. Some Wolbachia the strains are even able to directly reduce viral replication inside the insect through mechanisms that we may or may not know that much about. The World Mosquito Program, or WMP, actually uses a Wolbachia strain called WMEL. They all have names to infect Aedes mosquitoes or Aedes aegypti, and these mixed sex mosquitoes then infect the rest, as in like they infect males and females. They are intended to infect the local population either through the female line or by mating, and giving them transmission resistance so that they are less likely to transmit viruses. Wolbachia has also been identified or found to inhibit replication of chikungunya in Aedes aegypti, and it inhibits the secretion of West Nile virus in a cell line that was derived from Aedes aegypti. So it's not sure that it'll do the same thing in vitro, but it's cool that it can happen in situ. So this is the World Mosquito Program, and I think it's interesting how they're using Wolbachia and have this whole website for it. So you can look at that if you like. If you just search World Mosquito Program and Wolbachia, it'll come up. Excuse me. So I mentioned cytoplasmic incompatibility earlier. And some organizations are taking big advantage of this by creating opportunities for males or infected males to mate with uninfected females in the wild population, since none of them would naturally be infected. All of their eggs would become infertile. So by releasing infected males that don't usually carry Wolbachia, they would be able to temporarily reduce the mosquito population. And this has been conducted in, like this actual release of infected males has been conducted in Singapore and in the US recently. So this life sciences arm of Google, or Google's parent company, I should say, Google's parent company, Alphabet Incorporated. It's called Verily, the company, and they use this method and have released about 20 million Wolbachia infected male Aedes aegypti mosquitoes in Fresno, California, which is 3 hours away from the home I grew up in. And this is in an attempt to control or combat the Zika virus, because it was introduced to Fresno and the mosquitoes boomed in population. And then in 2017, also 2017, the EPA registered a company called Mosquito Mate Incorporated to release Wolbachia infected male mosquitoes in 20 US states and Washington, D.C. And I think it's funny because it's cool, but it's funny that they called their Wolbachia strain ZAP. So pest control, this all sounds very cool and it's been effective and it's working in Fresno, which is neat, but and Singapore, but it can be very complicated to make sure that these interactions are going how you think they're going to go, because the reaction or the effect of Wolbachia on an insect depends on the strain and on the insect itself. So in some cases, Wolbachia infection has been shown to actually increase single-stranded and double-stranded DNA virus infection, which is the opposite of what you might want. And even though Wolbachia infection can increase mosquito resistance to things like malaria, it can also enhance pathogen transmission. For example, in Culex mosquitoes, it increased arbovirus transmission and rates of infection. So word of caution always. That's kind of how it goes, I suppose, with human-mediated control projects. So all that aside, now we talk about the bugs. What does Wolbachia do in insects? I'm going to revisit this statement of this slide for the mechanisms of reproductive manipulation. We're going to focus especially on feminization and male killing, which I just realized is something I did not put on this slide. So I will elaborate on that later. But those are the two most well known in Lepidoptera. So I wanted to talk to you about that. So these are 4 of the reproductive manipulations from one of the review papers I mentioned earlier. And it's a good summary of good infographic of these 4. Feminization, that first one, results in genetic males that develop as females, and it can be found in true bugs, in isopods, and in butterflies. Parthenogenesis eliminates males from reproduction, and it can be found in mites, in Hymenoptera, and in thrips. And then male killing, and it actually eliminates infected males during development to the advantage of the surviving infected female siblings. And this is found in beetles, in flies, in Lepidoptera, in butterflies, and in pseudoscorpions. Then there's cytoplasmic incompatibility, which we've talked about already, and it prevents infected males from successfully mating with females that lack the same Wolbachia types. And this is in basically all of the groups that have already been mentioned, plus a couple new ones, like crickets. So feminization was first observed in a small pierid butterfly species, Eurema hiccabi, found in Asia, Africa, and Australia, speaking of pierids. And in this species, Wolbachia seems to interfere with the sex determination pathway. So it has to continuously be present and actively working in cellular mechanisms throughout development to complete the feminization. So if it's interrupted in any way, then the individual becomes intersex, which I think I mentioned that in my notes in a later slide. And then the insight into the mechanism of male killing as a reproductive manipulation comes from the lepidopteran host Ostrinia scapularis, which is a moth, or the Adzuki bean borer. Cytoplasmic incompatibility, we've talked about probably enough, but it was found in multiple lepidopterans, and since it wasn't first found in Lepidoptera, we're just going to concentrate on the first 2. So, like I mentioned, the removal of Wolbachia during development, probably through antibiotic treatment, results in intersexual development. So the individual can become like a mix of male and female characteristics. I forgot what that was called, but maybe some of you have seen some of those specimens before. It's very cool because it can make them really hard to key out. It's interesting. And then this mechanism is unknown, but work is still being done. And it's been postulated that this feminizing Wolbachia can lead to the evolution of new sex determination systems, such as shifts from females being heterogametic, as in like they have different chromosomes, XY, to male heterogamety. So in humans, we have women have XX chromosomes and men have XY. In butterflies, males have ZZ chromosomes and females have ZW, but the feminization of an individual by Wolbachia might lead to the development of different chromosomal patterns. Okay, sorry. I was just trying to mute to clear my throat. Male killing was originally thought to be another example of feminization in this moth because the all-female broods found in Wolbachia-infected Astrinia scapularis would suggest feminization maybe by Wolbachia. When the mothers were treated with antibiotic treatment, tetracycline apparently, to remove the Wolbachia, all of the brood were male. All of the brood was male. In the absence of Wolbachia, the genetic females died during larval development, whereas in the presence of Wolbachia, genetic males became feminized and died during the larval development. So it's still actually lethal feminization. It's just that it kills the males before they are developing into adults. This occurs during embryogenesis, and it's probably to provide more food for the surviving female siblings and ensure their survival so that they can their proliferation, they can spread the Wolbachia. And recent evidence indicates that some male killing Wolbachia strains can reach really high frequencies in their populations, resulting in changes in host mating systems to accommodate for the scarcity of females. So if it was something like a choosy female situation, it would then turn into a choosy male situation where now instead of the female being able to pick from a lot of males and mate with the highest quality male and the male is competing for her attention, it would be the opposite way around because now there's fewer males. So now I'd like to tell you about my work with Wolbachia. Briefly, maybe you could tell I'm pretty excited about it. I think it's a really fascinating system and organism, and I think it's neat that I get a chance to work with it after hearing about it so much in entomology classes. Some of you might remember this video. I think I had it in last year's presentation. I might not have, but this is what I work on. This is a strepsipteran parasite. Strepsiptera is an order of insects that parasitizes a bunch of other insects, and it's relatively small for an order. It's about 630 species, and they infect 35 families across 7 orders of insects. So crickets, katydids, bees, wasps, ants, mantises, cockroaches, true bugs, and silverfish, which is wild. And that video was me pulling Xenospechia, an adult female, out of its host wasp Polistes fuscatus. I caught these upstate in Black Rock Forest. And I did this so that I could isolate the parasite for whole genome sequencing. And we did do that. We sequenced it in-house here in the museum with a sequencer. And that paper is in the works. I'm just adding this last Wolbachia story to it and figuring that out before we try to get it published. But something interesting when we were doing quality assessments on the genome that came up was this is a blob tools plot. It's this program that you could run to assess the quality of your reads, but also the check for contamination, essentially, because this assigns each of your reads of DNA, each strand of DNA to an organism. And if you have a lot of contamination, then the genome's no good because it's not actually what you wanted to sequence. But as you can see, a lot of this is arthropod, which makes sense. That's what we wanted. And the clustering is pretty tight. So it implies that all of this is from one organism. But you can see that little splotch of orange assigned to proteobacteria. You know what's in proteobacteria? Wolbachia. So my external advisor, Dr. J.R. Nikathirathambi, got very excited and told me that I should start reading into Wolbachia's history in Strepsiptera. And it's only been seen a couple of times and not in Strepsipterans that infect wasps, which are the most widely known. So it's interesting that no one's been working on this. But what I did was I put the whole genome file into the MLST database to check if it had matches. And we came up with this profile. So all of these genes were present in the genome and they were Wolbachia. So those reads in that splotch of orange belonged to Wolbachia that were present in this Strepsipteran parasite. And this is the profile that it gave us. So those first three, FBPA, GATB, HCPA, those are 3 of the 5 typing genes, identifying genes. So my job was to check if its host and one other parasitized wasp had the same or any Wolbachia. And I'm realizing I put these in the wrong order. This should have been a big reveal in a couple slides, but basically, yeah, they did. And that was exciting. So they had its host, Polistes S1, the second graph, second table, matched with those same 3 type genes. And then this parasitized second wasp matched but was messy. So it's a less confident match in some respects. So this project had this kind of thought process, this thought tree going on, where we needed to establish that there was actually an association between Wolbachia, Strepsiptera, and the Strepsipteran host, wasp. So we had to go through like, is there Wolbachia only in the stylopized host or the parasitized by Strepsiptera hosts? And if we said yes, we would have to check if they're the same. If it was yes, but not all of them, there's the chance that the stylopized hosts were different strains or there's something else going on. And then if no, then Strepsiptera probably doesn't transmit Wolbachia in the system. So that was the first test, was to see if there's Wolbachia in the stylopized hosts. So I extracted DNA from the host abdomens. I did PCR targeting, which is polymerase chain reaction, which makes a bunch of copies of a certain part of DNA that you want. And I tried to amplify, I tried to make a bunch of copies of the 5 typing genes, the 5 identification genes. And I got them, at least for the actual host that the xenos, the Strepsipteran, came from in that video. And they were very clean, or they were relatively clean for me at least, on this gel, because I ran this, essentially I ran the PCR product through a gel to visualize the quality and the size of the DNA fragments to make sure it was actually what we wanted. If it was what we wanted, they should be around the 500 base pair mark, since a gel can also tell you size, and it was. I forgot to label that on this one, but you can see it in this next image. That's 500 base pairs. And excitingly enough, the other stylopized wasp came through as Wolbachia positive as well. This is a very grainy image, please forgive me. And the non-stylopized wasps had no bands, or bands where they weren't around the 500 base pair mark, so we could probably mark them out as not Wolbachia. So that was the result we wanted, or the result we were hoping for, even though you shouldn't really hope for things in science. So now we had to test if the Wolbachia is the same, and we could do that by running them through the database, which I showed you earlier. But having those sequences, I could also test that in a couple different ways. So I generated these haplotype maps, which are where I have multiple sequences from different individuals of the same gene, which are those 5 type genes, identification genes. In this case, we only had 3 because there were only 3 in the Strepsipteran genome. But these maps show how many differences, those tick marks are differences there are in the gene sequence. So AGTC in Xenos could be AGGC in Polybia, and that means there's two differences. So that's what that looks like. And all of these are showing us that the parasite genes of Wolbachia match up with the wasp host, the host wasp genes of Wolbachia, and they're different from Wolbachia genes taken from other organisms. So that was very exciting. FBPA is especially strong evidence because Polistes dominulus is a different species of host wasp in the same genus as our host wasp, and it had some differences. So even though the other two genes don't reflect that, it could be due to multiple factors, and there were some small differences, but they don't come out in this scale at the haplotype map. Other strong evidence was this phylogenetic tree that I generated with all of the type genes. So I combined the sequences of all of the genes, aligned them so that each difference, like each position in the gene sequences were comparable because they were all lined up. And we find that Xenos, which is our Strepsiptera parasite, is in a group with Polistes fuscatus, all of the stylopized host wasps are species, and even neater, Polistes, the host wasp, is nested within Xenos. So it's definitely like part of that grouping. And then Polistes dominulus is up here with these other closely related genera, showing its differences. So given all that evidence, we can pretty confidently say that Strepsiptera, at least in this system, probably transmits Wolbachia to its host, Polistes fuscatus. And that has a lot of different implications with how Wolbachia was spread in Hymenoptera and how it might still be spreading. And that brings up a lot of questions about what Strepsiptera is doing with Wolbachia, if it's helping or hindering, probably helping since it's present, but I can't say that. I don't know yet. So these next steps are to type and profile the strains. When we put them, those three identification genes in the database together, there's no profile for them yet, so they don't have a strain that they belong to. So we could establish that. We could find out which strain they might be part of. So I'd have to upload that data and do more phylogenetics perhaps. And then as another project that'll happen this summer, we're going to test more specimens for Wolbachia associations between the Strepsiptera and the host, seeing if they have if they have Wolbachia, and then if the Wolbachia is the same, just the same project. Specifically, I want to do this in Polistes canadensis. I caught 600 total specimens of that in Guyana in September last year. And we caught about 600 because I suited up in a bee suit and went into an abandoned building and knocked around wasp nests until the net was full. And we caught 11, perhaps, stylopized. So we want to check with this very robust data set if only those 11 have Wolbachia in them. And then additionally, we have a bunch of host parasite pairs. I have some on my desk right now that we want to test for Wolbachia. As a large-scale future project, we want to see if this is a host switching event, or if these are just kind of one-off infections, if the Wolbachia might be spreading perhaps through the population, but this is unlikely because strepsipterans, as far as we know, castrate their hosts chemically, but there's a chance that it might be spreading through contact within the nest or things like that. So that would take a lot more time in culturing and things because it's bacteria, and you have to culture bacteria in bacteria projects, I suppose. So that's all I've got. Please let me know if you have any questions. I should have included that video of me catching wasps because it's really dumb in Guyana. I think it's very fun. Yeah, I'll post this talk on my website, and I suppose the recording will be available, but I don't have the citations up yet. I will do that before I upload these slides. Thank you for listening.
Great. Thank you, RJ, very much. That was a great presentation. We are going to open for questions. So anyone in the audience, they can unmute and ask a question, or if for some reason you can't, you can also type a question in the chat. So we're going to go ahead, and if anyone wants to ask a question, now's the time.
I have a question, RJ. It's Joyce Clowley. You showed briefly a photo of there's a program to suppress mosquito-borne diseases in humans. And it looked in the photo, it looked like there were children in front of some sort of kiosk, and they were handing out what looked like little pink cups to children. And were those cups containing the Wolbachia infected male mosquitoes for distribution in their communities, or what did that represent, that photo?
That is a good question. I can't say confidently because I actually did not look at that closely.
Oh, there it is. There it is.
Yeah. Can you see it, Zoom?
Yes. Yeah, first I thought it was going to be Kool-Aid, but I don't think so. I think it's mosquitoes.
That makes sense, though. That would make sense, especially if they're even if there's liquid in that cup, it could be that they're showing them the nymphal stages of the mosquitoes that are infected. I think that would be very cool. It makes sense that they would show them those because that is what the World Mosquito Project does. But
yeah, I
I'm
didn't
sure
see the liquid, so I thought, oh, maybe they're handing out the male mosquitoes, and of course they don't bite, so that would be okay to introduce into a community or something. But yes, maybe it's just like a show and tell to see the early stage of the mosquito. Thank you.
Yes, of course. Thanks for the question.
RJ, this is Lee. I'm wondering, since these bacteria require a host, how do you culture them?
That's a great question, and one I'm also not confident in answering. I'm not a microbial person. This is my first foray into it, and it was brought up by one of my committee members that a reviewer might ask us to culture the Wolbachia to identify it if we went a bit further in our analyses, especially if we were trying to identify the strain. But from what I read while I was curating these slides, there was one statement about how some strains can culture and some can't. But I imagine it's difficult.
Yeah, stuff to figure out what exactly they need. Can I also ask, it's an intracellular or an intercellular bacterium or parasite?
Yes, I think it is intracellular.
Intracellular.
Yes, it is intracellular, so it has to exist within the cells of its host.
All right, thank you. It's very good, very interesting.
Thank you. Thanks for your attention. Thank you, I need it. I'm supposed to defend next year by summer.
Good luck. Good luck with that.
Thank you.
Sounds good so far.
I appreciate it. I see a question from Ellen in the chat. You're probably about to bring it up. The insects from Guyana are not alive. It is a long story, but there are customs agreements that we have to adhere to when we go to different countries to collect, as you might know. And this was an expedition for the museum, for the AMNH itself. It's the first expedition I was able to take part of, and it was a big thrill. But they're still in Guyana. So for the last couple months, we haven't been able to access them. governmental complications and red tape. But some of our lab, our museum specialists, our advisor, and one of my lab mates, they're actually all going to Guyana along with a couple other curatorial assistants to physically take the specimens and bring them back, because otherwise they won't be shipped to us. So we'll have them by this summer, but they're all going to be in varied states, we suppose. Mine were in ethanol when I left them, and we have collaborators in Guyana who have been tending to them and going in every week to top off the ethanol. But it's a hassle for these specimens. So I'm just hoping that since some of these specimens that I've been testing for Wolbachia have been pretty old or in degraded condition and still come out with Wolbachia positive results, that it'll be the same with those specimens from Guyana. Thank you for the question.
Last call for any other questions from the audience. Hearing none, I'm going to thank you again, RJ, for presenting, and thank you to everyone for attending the meeting today.
Of course. I'm going to very briefly ask, this is very silly, but right now I'm doing a social media feature for this organization called Pinoy Scientists, and I would like to take a very brief video of me waving with the screen in the background with all of you. I know that your videos aren't on, but can I do that really quickly?
Sure, definitely.
Thank you. Okay.
We
I'll
would
take it.
turn on the video.
Oh, if you like, thank you. Yeah,
Oh.
totally. That's very sweet. Here, I'll expand the screen a little bit. Okay. Here you go. I'll take it. I'll take it now. Thank you.
No problem.
I appreciate it. It's been very weird videoing myself all week. Okay. Enjoy your evenings. Thank you
All
so
right.
much.
Thank you, and thank you to everyone who attended. Have a very good night.
Thanks, RJ. Appreciate it. Next time, take a video with you and the wasps, okay? Thanks, Edmund.